Okay, good evening once again. I guess only the strongest students have left. Good. Before we start, let's say a small congratulation, because today we begin the 8th lecture, which means after I finish today's first part of the lecture, that will be exactly half of our course, so exactly 50%. So yes, I guess that's really great, and that's a good occasion to have, I don't know, some tea with chocolate cake or something like that. So that's really good. uh, One more thing, I guess. I checked today our statistics. Let me start the thing. Yeah, I guess our rate of turning in gets lower and lower. So we started with 19 works submitted, and now we are somewhere at 13 for midterm 1 and essay 1. So please, if you did not submit anything, please consider turning in some problems, because if we get this way, linear regression tells us that till the end of the course the number of submitted works will get to 0. So I don't think that's good. I think we should all get to the end of this course, because the course is pretty simple and nobody should fail. Good. Now we can get to the lecture. No, I didn't start one more program. Here it is. Good. Okay, now we are good to start. Okay, so today we start molecular signaling. The thing is that cells, mostly they constitute some sort of multicellular organisms, and of course in this case they live in sort of community, and they should interact somehow. So humans interact somehow, and cells should interact as well. Well, I guess even humans can be much better with solitude than cells. So what does it mean? First of all, we have specific cells that can sense physical stimuli. So be it mechanical, I don't know, some photons, so on and so forth. And they need somehow to process this information, or they need to pass this information further to some different cells. Many types of cells, they release particular chemicals that can influence behavior of another cell. These are some sort of extracellular signal molecules and or simply signals, and I will show you a few examples today and why do we need them. And of course, if we have some sort of organism that is right now developing, you know that all our bodies, they need some sort of precise architecture. So we need to create eyes in a head and not somewhere else, and so on and so forth. So everything should be precise. And how do cells, if we all start as a one cell, just one cellular organism, and then we transform in all of these in a multicellular organism, we should somehow cells should somehow communicate who will become an eye, who will become a hand, and so on and so forth. So signals are needed, and cells should react to these signals. And today we will consider some mechanisms of reaction to these signals and how everything works. The cell that receives a signal is termed a target cell. So you will often hear the term target. Typically, the signaling molecule binds non-covalently to a specific protein in the cell, typically called the receptor. How do you think? Why non-covalently? What would happen if these connection will be covalent? Remain permanent. Yeah, absolutely correct. So you don't want covalent bonds because covalent bonds are hard to disconnect. If you have non-covalent bonds, you can recycle that receptors later, and you can expect that that sort of molecules that connected to the receptor, and we call them ligands, so term ligand new for you, may eventually go away, and receptor will be deactivated. Okay, we get to that. Good. So release and reception of such signals is a fundamental process known as cellular communication. Good. Here is a generalized signal transduction pathway. What is this sort of pathway? It's a series of steps leading from initial detection. So a cell has detected some sort of signal that can be a chemical produced by another cell, that can be some sort of physical interaction. So a photon came, or I don't know, some mechanical action, whatever. And these series of steps lead to the final response. This whole thing is termed a signal transduction pathway. Signal transduction pathways, they ultimately lead to activation, or in some cases inhibition, of one or more effector proteins. So what are we acting on? We are acting on some sort of proteins. These proteins, they can be some sort of enzymes, for example, and that's why you can you can inhibit or activate some sort of biochemical pathway in the cell. So that's good. Interesting question for you. Here you see this sort of transduction pathway. Here you see a receptor. This receptor was able to detect some sort of signal. In this case, this is a ligand. I don't know why it's H, because I would rather draw L, ligand. It becomes activated, and then something is happening inside the cell. Look here. Here we have an effector protein. And here are some sort of intermediate chemicals or proteins that are activating each other, producing some other chemicals. These chemicals are activating some different proteins, so on and so forth. Why do we need all of these? Why don't receptors activate immediately the effector proteins? How do you think? What's the reason? You can see that from this image, like immediately. Amplification. Yes, absolutely correct. So this is a sort of amplification of a signal. Good. So there are essential features of most signal transduction pathways, and you have correctly said signal amplification. That's one thing. And another thing is some sort of feedback repression. So the thing is, you don't want your pathway to be permanently activated. So everything started, but it should stop at some point. So cells must be able to suppress or turn off a signal transduction pathway once the cell has adequately responded to the signal or the signal has been removed. So that's pretty obvious. Good. Let's look at signaling molecules and how do they act. They can act locally, they can act at a distance. Depending on how they act, you will hear different terms. So, for example, endocrine signaling. So you are acting at a very, very long distance. So, for example, hormone secretion. You secrete hormones into the blood by endocrine gland, and then some very distant target cells may detect that sort of hormone and somehow adequately respond. So there are many examples. I'm sure you can name on your own something around 3 or 5. Good. We can cells may signal on a very, very short range, for example by plasma membrane attached proteins. So two neighboring cells may signal to each other, well, about something. So maybe so-called paracrine signaling. So cells are pretty adjacent, but they are not in an immediate contact. So one cell, once again, may secrete some chemicals that will be detected by the another cell. And there may be some sort of autocrine signaling. So cell may secrete something that activates receptors on its own surface. This is pretty often seen in cases of different cancers, for example. So some cells, when they become cancer cells, they start creating some chemicals that activate certain receptors on their own surface, and this way they are promoting division and so on. Good. Here are a few examples of all of these cases. For example, endocrine signaling, where hormones act on target cells distance from their site of synthesis. Paracrine signaling, when signaling molecules released by a cell affect only those target cells in close proximity, so few micrometers. So paracrine, that's few micrometers. As example that you definitely know, a neuron releasing a neurotransmitter. So if you know synaptic cleft, one neuron is releasing some sort of neurotransmitters, and the other neuron should should react to these neurotransmitters. That's basically a paracrine signaling. Of course, there are different cases, but this one is probably the most well known to you all. Autocrine signaling, what I have already told you, many tumor cells release growth factors that stimulate inappropriate, unregulated self-proliferation. And some signaling molecules, they can act both at short and long ranges. So you should not consider that each molecule is some sort of predefined range. So it may depend. For example, epinephrine, also known as adrenaline. So it functions as a neurotransmitter, so that's sort of paracrine signaling, and that's some sort of few micrometers, so it just gets through the synaptic cleft through that few micrometers, and that's all. But it may function as a hormone for endocrine signaling, so very long range. The effector proteins in signal transduction pathways are often enzymes or transcription factors that induce two major types of cellular responses. So rapid, short-term changes, so we need to change something rapidly, and these activities, they change seconds to minutes, or some slower long-term changes from hours to days or maybe even years. So short-term changes. How do we make something really quick? So we should, first of all, we can modify some pre-existing enzymes. If you need some rapid change, and that change is expected to be pretty short-term, you cannot expect to synthesize new enzymes. That takes too many time. So too much time will be spent on synthesizing some new enzymes. So you are using some pre-existing enzymes and you are modifying them somehow, probably by phosphorylation or ubiquitination. So that depends. Phosphorylation is probably the most often seen way of modification of enzymes. So you turn it on or off or whatever. And long-term changes, these are typically changes in gene expression. So if you are changing something for a long time, it may have sense to activate or deactivate some genes, because now you have time to synthesize some new proteins and so on and so forth. So you can run that machinery for a long time. Of course, you don't expect that change to be immediate, because you need time for all that synthesis to go. But these changes can be pretty prolonged, and you may expect that whatever you have changed, you have turned on some gene that will make that will affect the organism for a pretty long time. These changes may include some changes in amounts and activities of specific proteins within cells, and they are pretty critical for cell proliferation, division, and organismal development. What else? Here are once again different cases of cell signaling in these slides, so you may check them once again. It shows that cellular signaling controls various aspects of multicellular life forms. So not only the key biological processes such as cell division, differentiation, growth, and cell cycles transition, but also specialized cell-specific functions such as neurotransmission, pathogen sensing, phagocytosis, and antigen presentation are controlled by specific signaling pathways. The process of autophagy and nutrient cycling and recycling are some accessory pathways that are triggered by definitive signaling cues. So, for example, insulin and glucagon, they are turning on and off certain pathways that we have already considered, like gluconeogenesis or glycolysis. So they are controlled with hormones. If you have ever studied, how do we call that? Well, okay, forget about it. The interaction between receptor and ligand. Just few words. So we have already considered that interacting through some sort of strong chemical bond is no good. So we don't want covalent bonds. What do we have? Which options do we have if we don't want covalent bonds? We have ionic bonds. We have hydrogen bonds. We have van der Waals interactions. So interactions due to some sort of fluctuations of the dipole moment. We have all that. We may have some sort of hydrophobic interaction. So if some part of the receptor is hydrophobic and some part of the ligand is hydrophobic, that will attract them together. Well, basically, our regulation of hydrogen. What is the question about hydrogen? Hydrogen bond. No.
I was just commenting on hydrogen bond. Yeah.
Yes.
It's not much. I didn't really ask a question.
Okay. Okay. Here is some sort of scheme, pretty schematic scheme, when we have a receptor R and ligand L. So receptor and ligand they interact and receptor is activated. So here is this sort of scheme. You might guess that we can write some sort of differential equations that describe this sort of interaction. Only the active conformation of the receptor R star can trigger the subsequent steps in the signal transduction pathway. So receptor should be activated to turn on that pathway. Thus, receptors are considered to be allosteric proteins. We have seen that in previous lectures, I guess. And as with all allosteric proteins, there is an equilibrium between R and R star, so activated and deactivated form. and the ligand binds much more tightly to the activated form, to the R star, than to the R conformation. Thus, it pushes the equilibrium to the right and activates the signal transduction pathway. Yeah, here is a neat catch for you. So receptor can get from the inactive to the active state on its own, just randomly. It may happen. So there may be some sort of basal activity of the receptors just turning on just because they want it. And nothing we can do because this is some sort of random process. But the probability of turning on their own is much smaller in the absence of the ligand than in the presence of the ligand. So that's some sort of chemistry. So ligand stabilizes the active conformation of the receptor, and that's why you have a large equilibrium shift in the direction of the activated receptors. But there's nothing perfect in this world, so you should know that receptors may turn on their own, just randomly. Of course, there will be some sort of mechanisms that don't let them transform these into some sort of a disaster, and there should be some number of receptors activated before the cell starts performing some actions, and the more receptors you need to activate to start some pathway, of course, the lower is the probability that this activation was just some sort of a chance. So the higher will be the probability that they are activated because a ligand came, not just on their own. Good. Here is one more, a nice picture for you that depicts many different types of receptors. I guess we will get through, well, I'm not sure if all of them, but many of them when we'll get through this lecture. But just some sort of a reminder or something like that, that we can classify receptors into two broad categories. One is the cell surface receptors, and I guess we will spend a lot of time talking exactly about these receptors because they are pretty interesting for us. But of course, there are intracellular receptors. Some of them may reside in nucleus, so there are some nuclear receptors that activate certain genes and so on and so forth. I guess we'll see some of them, so please remember that receptors can be anyway. I will probably focus mostly on the cell membrane and on the receptors that reside on the cell membrane and are getting some sort of information from the outside world. But there are certain receptors that can be in cytosol, that can be in nucleus, and so on and so forth. So they can be anywhere. Moreover, some chemicals, even coming from the outside, they can activate some receptors that are in the inside of the cell. For example, steroid hormones. They act on the nuclear receptors in the nucleus of the cell. And here is, I guess here is an interesting question for you. If I have two chemicals, one is hydrophilic, the other is hydrophobic, how would you expect which one of them will preferentially target some sort of surface receptors? So suppose they both carry some quite important information for the cell, so the cell produces receptors for these chemicals. So for one of the chemicals, it produces receptors on its surface, so a cell surface receptor. For the other chemical, it produces a receptor, let's say in the nucleus, so inside the cell. Which one is which? So which one do you expect to activate the surface receptor, hydrophilic or hydrophobic, and which one do you expect to activate the nuclear receptor, hydrophilic or hydrophobic? hydrophilic for nucleus. Hydrophilic means it likes water. Guys, you are pretty close, but I guess you are a little bit mixing some terms. So hydrophilic substance likes water, so it really good so when dissolving in water and so on. And hydrophobic does not like water, but it may dissolve in something lyophilic, in some fats or so on. Yes, so nuclear hydrophobic, surface hydrophilic. That's absolutely correct. So why? Because to get to the nucleus, you should pass through the membrane. And what is a membrane? That's a lipid bilayer. So to get through the membrane, you should be able to somehow dissolve in that membrane or something like that. So to be able to cross the membrane, you should be hydrophobic. And if you check, for example, steroid hormones, you will see that they look like cholesterol that is hydrophobic. Well, they are basically synthesized on the base of that on cholesterol, and I think I even have some sort of schemes in this lecture. But we'll get to that, I guess, tomorrow, not today. Good. So here is some sort of reminder about all these receptors. So we have some nuclear receptors, we have some surface receptors, and we may have some receptors in the cytosol. So that's pretty possible. Here is a small reminder for you. Because many target proteins we will turn on or off by adding or removing phosphate groups. So here you see this sort of example. Well, it tells you that protein is activated by adding phosphate group, but it's not always so. So there are some sort of exceptions, and well, it's biology, it's not math, so you might expect some exceptions in pretty all cases. So protein kinases, they add phosphate groups to specific residues of target proteins. So protein kinases, they will add you a group, and protein phosphatases, they remove phosphate group. So when you see these two names, you should more or less remember whether they add a group or do they remove that group. The catalytic activity of all kinases is highly regulated. Commonly, it is modulated by the binding of other proteins, so they don't act on their own in most cases, and they are modulated by something else. And we will probably see what else today. Okay, here are a few more examples. So, like enzyme binding to a substrate, each type of receptor binds only a single type of molecule or a group of very closely related molecules. We will see examples. A single ligand may stimulate different cell types to respond in distinct ways. So it doesn't mean that one ligand just makes one action. No, different cells may respond differently to one and the same chemical. Here are some sort of examples of acetylcholine. In skeletal muscle cell, release of acetylcholine from a motor neuron innervating the cell triggers muscle contraction. In heart muscle, the same chemical reduces the heart rate. In pancreatic acinar cells, it triggers secretion of digestive enzymes. So the same chemical, different results. Sometimes the same receptor found on different cell types can, in response to binding the ligand, activate different signal transduction pathways leading to activation of different effectors. That can be, example, beta-adrenergic receptor for hormone epinephrine, depolymerization of glycogen to glucose in liver and muscle, but in fat and adipose cells, it will activate hydrolysis and secretion of stored fat, so-called beta oxidation of fats. I guess at some point we will need to get to that. Here is an example of signal transduction in case of some organism developing. So here is some sort of experiment, and gradient of the paracrine factor activin, a morphogen, causes concentration-dependent expression of differences of two genes in unspecified amphibian cells. So here you see some example where we have some beads with no activin or beads that contain some sort of activin. And we see that it diffuses, some gradient is created, and we see that and we can measure expression of certain genes that we are interested in, and we see that these genes are expressed differently. So a threshold value appears to exist that determines whether a cell will express X-bra or gene gusicoid. I'm not sure how to read that, so it's really a strange name for a gene, but we have what we have. So depending on that gradient, you see that here is expression threshold for one of the genes and for another of the genes. And if you have turned in some cells, you have turned on one gene in another cell, you have turned another gene. You may expect that these cells now may develop differently, and one will give you, I don't know, some eyes, other will create you a leg, and so on. Well, of course, I'm a bit oversimplifying, but you get the idea. So concentration and gradient of certain chemicals during development may define which genes to turn on or off in different cells, and depending on that genes you have turned on or off, you will get different ways of development for that cells. So that's how it works. Okay, a little bit on kinetics. Very similar to Michaelis-Menten kinetics, and if you look at these images, you can pretty guess why. So we have almost the same equations. The only thing that changed is the meaning of the letters. So that's all. So we had some enzyme binding to a substrate and creating a product. In this case, we have a receptor binding to a ligand and created some sort of activated receptor ligand complex. And if we do all the math, you will get pretty similar result for some sort of constants. So we had a Michaelis constant, Km. I hope you remember that. Here we have Kd. It is called a dissociation constant. And you can guess that this is the how much ligand you should have, what should be the concentration of the ligand to bind 50% of the cell surface receptors. And here is even some example for you, just to give you some feeling of numbers. So Kd for insulin in liver is 1.4 times 10 to the minus 10 moles per liter. So 10 to the minus 10, that's a really tiny number. So the normal concentration of insulin in the blood is about 5 times 10 to minus 12. And after a meal, 2.5 times 10 to minus 11. So you might guess that much less than 50% of the insulin receptors are activated even after you have eaten something. Well, pretty reasonable. Why do we need to activate them all? Probably just a few are enough to trigger the pathways we need. Here is the plot. You see it's pretty similar to the plots we had when we considered Michaelis-Menten kinetics. So you might guess the same formulae, pretty similar kinetics, just some different numbers and different meaning. Well, I like these formulae for one more reason. They show you, let's say, power of math. Because when we consider the problem of enzyme kinetics and the receptor ligand binding, from the biological point of view or even some physical point of view, they are pretty different because you have pretty different processes that you are considering. But when you get to the formulae, when you remove everything that is not needed for the kinetics, you just leave out the bare problem that you are considering, it turns out that you get the same formulae. Absolutely the same. So for the mass, it doesn't really matter. So I think it's a really nice point to note that in many cases you get pretty similar formulae for pretty different processes. So only when you create some abstract version of your problem, so you have some problem, it looks very different from many other problems, but when you abstract that, when you remove everything unnecessary and you leave only these things that you can write as formulae, it may turn out that it is very similar to pretty different problems. They all collect together and give you some overall view that you could never have just looking at each particular case. So I think it's really it's really nice. Good. One more thing to notice, that all these receptors, even on the surface of the cell, they are not static. And by these I not only mean that they are constantly some sort of activated, deactivated, well, because something is happening on the outside, but they are constantly recycled and synthesized de novo. Why does the cell do that? Well, the cell basically acts so to respond to hyperactivation or to to hypoactivation. So if we have a lot of signal, so here you see an example when we have some sort of effect from the receptors. Some are activated by the nearby neuron, some others are activated by some sort of different chemical. So something, I don't know, cocaine, for example. They are hyperactivated. The cell is seeing that it gets constantly some sort of signal, and it starts to down regulate. So a new word for you, down regulation. So it down regulates the receptors, in this case by recycling some of them and just leaving just a few. So to somehow to get away from that hyperactivation, so to not be constantly activated, because that doesn't bring any useful signal, it's just permanent activation. Or it can up-regulate, so word for you, up-regulation, and synthesize some new receptors, because, for example, old receptors don't get any signal for whatever reason. In this example, you have some sort of antagonist that has that is connected to that receptors and just does not let the signal to pass through. So they cannot bind now to the molecules they were initially designed for. And the cell upregulates receptors by synthesizing some additional receptors that now may react to this signal. So okay, that's for you to remember. Good. Some sort of very short interlude, pharmacological. Of course, we don't have time to get through all the pharmacology, and of course, I don't know so much to give you the full-fledged course, but probably some terms for you will be good to know. Because if we think about different drugs, they are most cases activating or deactivating certain receptors. So to start or to stop some sort of signaling pathways in the cells. So if you check, for example, I don't know, drug bank. So if you check drug bank, there is a large list of different drugs, and for many of them, you can find there some sort of references to articles or even short descriptions of the mechanisms, how do they work. And you will probably find out that many of them are activating or deactivating certain receptors. So yes, it's not just a random insertion, and it really has to do with whatever we are considering right now. Good. So you see that all that pharmacological thing we can divide in two parts: pharmacokinetics and pharmacodynamics. So pharmacokinetics, that's how the drug behaves from the moment you have administered that drug. So it may be you may have eaten some sort of pill, or you got some intravenous bolus, or whatever, many different methods to get drugs inside, but you did that. So drug for some time it lives in systemic circulation. So here you probably should consider if that was a pill, it should be the first pass through the liver, and you will hear words like bioavailability and so on and so forth. But at some point it gets to the site of action, and then it interacts with receptor. So we will be mostly interested in pharmacodynamics because this is the part where interaction with the receptor is performed. And then it interacts with the receptor and you get some sort of response. So something happens depending on whatever. Here is just a small slide that shows you that here we have some drug in a tablet. It gets through the liver, it gets into your systemic circulation. And then there is some sort of drug metabolism. It is partially excreted, partially processed somehow, so whatever is happening. So here are just a few terms for you, well, just to know. So minimum effective concentration is the lower boundary for effective drug concentration. So plasma concentrations below MAC have a high probability of being subtherapeutic, so they do nothing. Maximum tolerated concentration, MTC, is the upper bound for optimum drug concentration. So if we have something higher than MTC, it will be probably toxic, so we don't want that. The onset of action, these terms means that it estimates as a time it takes for plasma concentrations to reach MAC. And duration of action of a drug may be estimated as the time during which plasma concentrations remain within the therapeutic range. So here is some plot for you. You see a drug, so you have eaten some tablet, some small time passes, you get to this point, which means that you have reached MAC. The time from 0 to this point, that's what? Who can tell me? That's onset of action. It's onset of action. So it's time that it takes to get to MAC. Then for some time we have something like this. We don't reach MTC. We don't want our patient to die. Yeah, so we don't want this chemical to be toxic. And then it goes down, 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 and at some point it crosses MAC once again and goes to the subtherapeutic. So after this time, so here we have some sort of duration. This is duration of action. After this point, drug is no more effective. So maybe you need one more pill, maybe something else, whatever. And this is probably the reason, you might guess, why often doctors ask you, if you take some drugs, to take them at some appropriate time. So they tell you 12 o'clock, take this, and then 6 hours later this, and so on and so forth. So that's basically, that plot is basically the reason why they do it. And in some cases, well, not always, in some cases we defy the rules, but there are some cases that we need to follow the rules pretty strictly. For example, with antibiotics. If we don't follow the rules with antibiotics, we get problems because due to the evolution and changes in bacterial genome, we might get some bacteria that can tolerate the drugs that we consume. So we really don't want that, and we need to keep that level of the antibiotic within the range between minimal concentration to do something at maximal concentration before it gets toxic. So that's really crucial. And yeah, so other drugs that depends, but with this I should tell you certainly you should follow the prescription and timings. Good. Different chemicals activating receptors. So here you see a list of let's say classes of chemicals with respect to certain receptor. So what do they do and what happens? For example, a full agonist, that's a ligand or drug that is capable of binding and activating a receptor with maximal efficacy. Example, epinephrine and adrenergic receptor. So that may be the chemical that is synthesized within the organism, or that can be some sort of external chemical that does not exist in the organism, but you have somehow chemically created. That depends, but if it is able to activate receptor to full strength, that's full agonist. Partial agonist, you guess what it is. Activates receptor, but not so strongly. Example, buprenorphine and mu opioid receptor. And we consider that partial agonism, in this case, we consider that as good because that decreases the risk of the overdose. That's good. Inverse agonist, a drug that has the same binding site as a full agonist but reduces activity in receptor. Example, antihistamines and histamine receptors. Antagonist is a ligand that blocks the activation of the receptor. So it does not let receptor to activate by preventing agonists from binding. And in contrast to inverse agonist, an antagonist does not have any intrinsic activity on the receptor itself. While inverse agonist is a drug that has the same binding site as the full agonist, but reduces the activity in the receptor that would otherwise be constitutively active. So the example that I told you, that we do not expect all receptors to be deactivated and activate only when some sort of ligand comes. They can be activated randomly. That is some sort of basal activity. And inverse agonist is capable of making that activity lower. So when we have some sort of agonists, they are stabilizing the activated state of the receptor, and they are shifting the equilibrium towards activation. When we have inverse agonist, it shifts equilibrium towards the inactivated state. And if in norm you have some sort of basal activity of the receptor, they are sometimes just turning on, this chemical will make them turn on even with lower rate than without anything. And here are plots that show you what happens when you have these different sorts of drugs and depending on their concentration. So here we have low concentration, here we have high concentration, and here is what happens. So here is a receptor response, this is some sort of basal rate, and if you have full agonists, you start activating receptors. If you have partial agonists, you start activating them, but not so vigorously. If you have inverse agonists, you are going even down compared to the basal rate. And if you have antagonists, well, antagonist only prevents from binding with agonists. So if you don't have anything else except the antagonist, well, the basal rate is kept. If you have some mixture of antagonists and agonists, well, you will see the difference, and in this case, you do not. Here is one more image for you. Maybe this one will be even better for the clarification, here you see the equilibrium between inactive receptor and active receptor. Here you see how different chemicals are influencing this sort of equilibrium. So here you see the basal rate. So we don't expect all receptors to be turned off. There is some sort of basal rate. If we add agonists, full agonist or partial agonist, we have this equilibrium shifted to the right, to the active receptors. In full agonism, it's a large shift. In partial agonism, well, it's somewhat shift. If we have inverse agonist, you see that basal rate, basal activity is shifted to the left, so to the inactive state. That's what we have. Good. Here are examples for you. Here is epinephrine and two chemicals, isoproterenol and alpranolol. Well, you see that these chemicals are pretty similar to the epinephrine. What do? Isoproterenol is an agonist of the epinephrine receptor, so it activates the adrenergic receptor. It is used on bronchial smooth muscle cells and helps opening air passages in the lungs. So it may be used in treating bronchial asthma or chronic bronchitis and emphysema, so quite useful chemical. So it activates something. And here you see alpranolol. This is an antagonist of beta adrenergic receptor present on cardiac muscle cells. So you see some chemical difference between them and so on. And if you have ever heard the term beta blockers, so this is an example of beta blocker for you. Good. What else? Here is some sort of model for you. You see this equation. Here is some sort of response depending on different levels of drug. So this is a model, but it somehow works, and maybe in some cases you will meet this sort of equation and even be able to calculate something with that. So here you see the description of all these factors. I know that there are no I indices here. This means, how would I say that? Well, this equation is after you have performed some sort of regression. So this is for the eyes patient. So if you have many patients, these are some sort of points on the plot. So when you plot some sort of scatter plot, and this equation is after you have somehow fitted this kind of curve into that points, and you get something this smooth. So that's why you don't have that indices I here, but you have them in here. Maybe I should have fixed that, but I hope you will understand it anyway. Okay, so what about this model? So you see here E0, that's basal effect, so we expect some basal effect from the receptors, and we expect some sort of maximal effect. Well, effect is anything measurable you can get from your organism under consideration when you are, well, whatever, it may be anything, some activity of certain enzyme, for example, or something else, some motility of some cells, or whatever you can measure and transform into a number. Many of them will fit into this model. It has 4 parameters, so E0 like basal effect, Emax some sort of maximal effect that can be attributed to a drug, ED50 that's the dose that produces half of the maximal effect, and N that basically is called Hill factor. So that's a slope factor that measures sensitivity of the response to the dose range of the drug. We have seen pretty similar curve when we consider hemoglobin. There was some sort of reaction on the oxygen. Of course, we didn't call oxygen a drug, but if you abstract once again, you will get pretty similar model for the for that case as we have in this. So Emax model predicts maximum effect of a drug. So that drug has some maximal effect. It considers a zero dose effect, and it follows the law of diminishing returns of higher doses. So you see that this part almost nothing is changing, so you give some higher and higher and higher dose, but the result is, well, you get some result, but it's pretty small. So that's the law of diminishing returns. And this model is good because its parameters are readily interpretable. So if you have some data, some measurements, so you give some drugs, I don't know, to mice, and you have measured, for example, speed that they have when they run through some sort of maze, for example. That's pretty measurable characteristic, and you can try to fit it into this model, and maybe you will see that some drug makes them run faster. Why not? Here are some mechanisms. So here is a mechanism of receptor antagonism. Here is A agonist and I antagonist. I, it probably comes from the word inhibitor, and we will call that in case of enzymes. You see, once again, it's pretty similar case, but here we call it antagonist. And it's pretty good that we use different letter because both agonist and antagonist start with A, so having two A's in equations that mean different things would be pretty bad. And here is example of a competitive inhibitor or competitive antagonist, and irreversible inhibitor or irreversible antagonist. And here you see how that works when we have all these curves, so dependence of the effect. So let's say how many of the receptors are activated depending on the concentration of the agonist. Well, pretty measurable effect, so we can measure that. Well, as an example, you can measure something downstream the signaling pathway, but let's say we measure immediately here the number of activated receptors. And these are the curves that you have in case of competitive inhibition, and this in case of irreversible. So in case of irreversible, you are just removing part of the receptors. So here you see how that happens, and here are some equations that more or less describe what is happening. What else? Allosteric modulation. So a drug may bind at a site distinct from the orthosteric site, and it can alter the receptor's affinity for the orthosteric agonist. So orthosteric site, that's the primary site where we expect the natural ligand of the receptor to be bound. So there is some place where that ligand binds, and we know where it is, and we call that an orthosteric site. So we have already considered drugs that bind in the same site. For example, some sort of competitive inhibitors, they bind in the same site and they just don't let the ligand to get through and bind. But we can create drugs that bind in different sites. They are called allosteric modulators, and you can have PAMs or positive allosteric modulators and NAMs, negative allosteric modulators. Here are some examples like cinacalcet is PAM for calcium sensing receptor, benzodiazepines and barbiturates are PAMs for GABA receptor. Do you know what is GABA? That's gamma. I see. Yes, that's neuromediator. Yes, that's neuromediator. Absolutely correct. That's gamma aminobutyric acid, and that's one of the most important neuromediators and neurotransmitters. And I guess we will meet these sort of neurotransmitters in some of the subsequent lectures. Well, if you have read syllabus, you probably know that I have some sort of feelings to the neurobiology and neurobiochemistry, so probably we will have much more of that neural stuff in the future. Okay. Here are some more examples like meraviroc, that is NAM, blocking the binding of the HIV protein to the TCR5 chemokine receptor. So that's one way to, let's say, control somehow HIV infection. uh, ty- Ticagrelor and cangrelor are NAMs blocking the actions of ADP binding to the orthosteric sites on platelets, P2Y12 receptors, and so on. It has to do with blood clotting and so on. So we have different chemicals, different drugs that act on the receptors, and you see that there are different pathways to act. So they may activate receptors, they may prevent receptors from activation by different ligands, or they can even somehow reduce the basal activity of the receptors. So that depends. They may act, well, most cases they will not bind covalently, so that will be some sort of reversible process, but in some cases we can get even irreversible processes as well. Good. Now we get to the part with different receptor types. Uh, there are there are pretty many receptor types, and I will try to show you at least some of them, maybe the most prominent, and some pathways that are associated with these sort of receptors and pathways. Good. Ligand-gated ion channels. I will not spend much time today on them because we will probably have a lot of time to consider ligand-gated and voltage-gated ion channels and ion channels in general in the lectures when we get to something closer to the neurobiology. So we will have a lot of time for that. I'll just say that they work on timescales of milliseconds, so it's really easy to change something with these receptors. Why? Because they are letting in or out ions, so you have some sort of influx or efflux of ions in the cell, and they change the difference, the potential difference between the inside and the outside of the cell, and basically that that is a very fast process, and that allows to communicate some sort of information pretty quickly. So we will get to that. G protein coupled receptors. They work on scale of seconds, and I guess we will try to consider at least partially them today, and maybe we will finish tomorrow. So a large class of receptors and many drugs, they act by activating or deactivating these type of receptors. So it's really good to consider them. Kinase linked receptors. So these are receptors that have some sort of kinase properties. I guess insulin receptor is in these falls in this class, so that will be considered, I guess, tomorrow. And nuclear receptors that activate or deactivate some genes. So you have some more transcription of certain more transcripts from certain genes or less of them, and we will get to that as well. You might guess that these sort of nuclear receptors, they have a really large time scales, so for example hours or days or even the whole life. So that depends. And one of the example, let's say estrogen receptor. So here you see cytokine receptors. They work on scale of hours and so on. Good. Here is a small table with these types of receptors. Here you see some some small information on them condensed. So where are they? So what is the effector? What is the coupling? And some examples of these receptors, and even something about their structure. So you can consider this table and get some ideas of what they are. So here are small pictures of these receptors. So these are ion channels, and these are once again G protein coupled receptors. So they are somehow coupled to G protein, and Rank and Dale. Yeah, it may be Rank and Dale. I used some pharmacology book to get certain pictures, so yeah, it may be from that book. I need to check that, but I guess yeah. Well, I use so many books to steal pictures from there, so it's really hard to remember where did I get this all from. Okay, so here are some pathways how a signal can be transduced inside the cell. So that's the point I'm getting more to the surface receptors. So here is some, it may be through so-called G protein. I really hope we will get to that today. It can be some receptor activation of phosphorylation cascades, and that may be some conformational changes that open ion channels. So how do we change something? That's the way. Good. Really quickly getting through ion channels, because I guess we will have a lot of time to consider them later. So this is the fastest receptor in the Wild West. So they do everything pretty quickly. The changes in gradient and electrostatical potentials are pretty quick. What they do, they control the flow of ions, mostly sodium, potassium, calcium, and chloride. This changes membrane potential, triggers electrical or chemical responses. So they are used in many cases. So not only synapses and neurons, you can find them in many places. I don't know, maybe the most strange, let's say, when a zygote is created, so when a process of fertilization goes on, you have a rapid change of the membrane potential of the cell that basically blocks all the other spermatozoids to get into, so only one can get. There are different processes, many different processes, but one of the first and the quickest process that blocks everything and starts that sort of rejection that no other should pass is basically based on the change of the membrane potential. So not only neurons, we have many places when we need some sort of quick response, and most cases changing of membrane potential will be used. And they are targeted with many drugs, so benzodiazepines, anesthetics, and so on. Here are some schemes of these receptors, and you see that there are different types of these receptors. They can be ligand gated, so they react on certain chemicals. They can be voltage gated, so if you have change in membrane potential, your receptor may react to this by opening or closing. So that, for example, drives action potentials and calcium signaling. I guess we will see that in case when we consider muscular fiber, for example, or action potential in neurons. And there are so-called metabotropic or indirectly gated that are controlled by GPCRs or other second messenger cascades. like cyclic IMP or inositol 3-phosphate and so on. They are slower but have more long-lasting effects. So here are some schemes of these receptors. I'm not going into details really well because we will have time for that in the future. Just for you to know that the quickest receptor is some sort of ion channel that can be opened and that changes membrane potential. And you may need that membrane potential to signalize something very, very quickly. So you need something to be done immediately. In most cases, we think about neurons, but many other situations may occur when the cell would like to do something like that. Good. And it's really good that we still have some time, and I'll be able to at least start talking about GPCRs. So, why GPCRs and why do we spend so much effort and time on them? The human genome encodes something like 800 functional GPCRs, so G protein coupled receptors. That's 4% of the all identified human proteins. So you might guess they are important. They are pretty important because 4% of our proteins are basically GPCRs. They respond and detect many different types of signals, including odorants, testins, neurotransmitters, hormones involved in glycogen and fat metabolism, growth factors, and even physical stimuli. So the fact that we see right now something, we see computer, we see the screen, at least I hope that you are looking at the screen and all the slides I'm showing to you, that's because we have GPCRs, so G protein coupled receptors, and I guess I have some lecture at the end of the course about seeing, about how do we process, well, not process, but how do we react to that stimuli, to photons, and we will see these sort of receptors there. So they play a crucial role and in transduction of that signal and in reacting to the light. Approximately 35% of all drugs used in humans are agonists or antagonists of GPCRs. So once again, many drugs that we do, 1/3 of all drugs have to do with GPCRs. So if you want to do something medical, you will definitely meet with GPCRs and with drugs that affect GPCRs. So you might guess that these receptors are really under the heavy, let's say, under scrutiny of many people, and we are trying to understand them as good as we can. Okay, so what means GPCR? So that's G protein coupled, which means that our receptor is coupled to something, to some entity that is called a G protein. So what is this G protein? That's a heterotrimeric guanine nucleotide binding protein. So they are signal transducers that communicate signals from many hormones, neurotransmitters, chemokines, and autocrine and paracrine factors, so whatever. The extracellular signals are received by members of a large superfamily of receptors with 7 membrane-spanning regions, so they have pretty similar, they look pretty similar. Maybe, I'm not really sure, but maybe evolutionarily they were created from one receptor just by duplicating that gene and modifying that. That may happen. So we know that evolution sometimes creates some copies of genes and then modifies that copies and we get something functional. Good. Here are examples of different chemicals that act on receptors that later activate some G proteins. We have different, let's say, classes of G proteins. Here you see these classes like GS, GIO, GQ, and then these G proteins, they may activate or inactivate some enzymes downstream. I guess I will consider a little bit more details in the next slides, but here you see the connections. So GS may activate some metabolic enzymes and some ion channels, while these GIO, they activate some ion channels and transcriptional machinery and so on. So here is a nice picture for you. You can look at that and get some idea about these classes. All GPCR signal transduction pathways, they share some common elements. First, a membrane-embedded receptor that contains 7 membrane-spanning alpha helices. So that's a common property in these receptors. A heterotrimeric G protein, which functions as a receptor-activated switch by cycling between an active GTP-bound and inactive GDP-bound state. A membrane-bound effector protein. So I will show you, I guess, later a scheme. You will understand why that's membrane-bound. And proteins that participate in amplification and desensitization of the signaling pathway. So protein kinases and second messengers such as cyclic AMP and calcium, they often participate in the GPCR-mediated signal transduction pathways. So these pathways usually have short-term effects. They are not so quick and cannot be so short-term as, for example, for ion channels, but they are pretty quick. So that's the next level of the quickness. You might check the numbers if you return a few slides previously, and there we had some sort of table that shows you that time scales for different receptors. Good. So these pathways, they allow cells to respond rapidly to a variety of signals, so be it some physical signal or some hormonal stimuli, so whatever. Okay. So here is one more example of a G protein coupled receptor, and one more reminder that they have these 7 transmembrane helices. Human GPCRs are divided into several families. Members of each family are especially similar to one another in amino acid sequence and structure. The families differ from each other in details of how ligands bind to the receptors. So there are some subclasses, and I'm not sure if we need to know that, so just if you want to get really deep. Here are examples of 3 principal classes of G protein coupled receptors that bind their ligand in different ways. So here you have family A. Here you see this blue blue ball. That's what actually has bound to the receptors. So these are biological amines, or they can react to light or odorants or whatever. Here you have family B. It has this large domain outside, so ECD means extracellular domain. So it's part of the protein that is floating somewhere outside the cell. Well, not very distant, far from the cell, because you understand you can't. You will not make a receptor with some part floating emitter, but you get the idea that this part is connected to the 7 transmembrane helices by some sort of unstructured region. And here you have family C that reacts to biological amines or glutamate or calcium. So these are sort of dimers. Good. Here I have some examples of these families just to show you, let's say, nice pictures. Here they show you some inactive receptor. This is G protein. So this is G protein. It is heterotrimer. So these are in different colors. You see three different parts of this protein. So they are called alpha, beta, gamma. Here you have this receptor activated. So you have ligand that is bound to the receptor, and now the activated receptor can bind to the G protein. So G protein coupled. So now this receptor has somehow interacted with the G protein, and I will later show you what happens to the G protein. This alpha subunit, it will go away, and well, it first will exchange GDP for GTP, then it will go away, activate there something, I don't know, some maybe some cyclase that will make more cyclic AMP and so on, but here you get the idea. And this family A is the largest family in mammals. Segments of several membrane-embedded alpha helices and extracellular loops from the ligand binding site are shown. The site is exposed to the exoplasmic surface, but can extend down towards the middle of the membrane bilayer. And here is some example of the beta adrenergic receptor that reacts to the epinephrine. So here you see the epinephrine that interacts with the receptor. Once again, no covalent bonds, only some sort of hydrogen bonds, hydrogen bonding. Okay, this is the glucagon receptor. That's a member of family B. So that was family A, here is family B. Of course, GPCR. So here you see how that looks like. Here you see glucagon itself. Oh, I'm trying to draw red on red. Probably not very smart. So here it is. So here you see that, and it's bound to the receptor. Receptor changed its conformation, and once again it will interact somehow with the G protein. Okay. And the glutamate receptor, that's an excitatory neurotransmitter receptor in the family C of GPCRs. So we have considered AB, and here is a family C. So here you see that sort of dimer. So the long extracellular segment forms a sort of venous flytrap domain, and it is called that way, venous flytrap domain. Yeah. That comes from the plant venous flytrap. So it reminded to the researcher, that form reminded the researcher on the venous flytrap. And many members of this class of GPCRs, they dimerize through their venous flytrap domains. And here you see a small table that shows you how they activate and some sort of ligand binding and so on. So here you see example of the active and inactive receptor, so how that dimerizes during activation. Good. So how much time do we have? We still have 5 minutes. Let me please check where we are, so I can find a good place to stop. Because we have okay, we will get to deactivation probably, or to this. Good. Good. I guess this slide may be one more, and that will be a really good place to stop, and tomorrow we will continue. Good. So here is an example of the pathway. So what happens when we have some agonist binding to the inactive receptor? So here you see agonist binds to the receptor. Receptor is activated. Now you see this G protein. So you see that G protein is in some sense tethered to the cellular membrane. I have some slides, I guess, later that show this structure in a little bit more detail, so probably we'll consider that with some more details. But anyway, now what happens? They connect, and G protein, it has these three subunits, alpha, beta, and gamma. In alpha subunit, there is bound GDP, and after the receptor activation and binding with the G protein exchanges GDP for GTP. Then alpha subunit may go somewhere and activate something. For example, some effector protein. It may activate that effector protein. So here you see the example. Why ligand creates on slide 11? Yes. Binds. It shifts the equilibrium by binding the receptor because it stabilizes its active conformation. How do I put that? So if you have a receptor, so you have receptor in inactive state. Something may happen and this receptor turns on. Sometimes passes and it turns off. And these process, so no ligands, nothing. So we have just situation with solely receptors. They come to some equilibrium. So there is some probability for the inactive receptor to activate, a really small probability. Let's say of all the receptors, there is, I don't know, 1% probability that during the next second it will activate and get from this state to this state. And there is some probability that active receptor will deactivate on its own. And this is some high probability. So let's say there is a probability of 99% that during the next second the active receptor will deactivate. So what it means, there can be some sort of equilibrium between active and inactive receptors. So if I have 100 inactive receptors, during the next second probably one of them will activate. If I have one active receptor with a very large probability, almost 100%, during the next second it will inactivate. So I can do the exact numbers, but you can guess that if I have 100 inactive receptors and if I have one active receptor, that's, let's say, pretty close to the equilibrium. So each second one of these 100 will get activated, one of them, and this one almost certainly will get inactivated, and we still get the same numbers. So this one was activated, so number gets down to 99, but this one that was active inactivates, so we get once again +1, and here is our equilibrium. So is this part at the moment without any ligand, is it more or less clear?
Yeah.
Good. Now the ligand comes. Ligand stabilizes the active form of the receptor. So we have some ligand. What this means, now the probability of the active receptor to deactivate drops down from 99% to, I don't know, let's say 50%. These are pretty random numbers, but I hope you get the idea. So it stabilizes this state. Now what this means, if I have 100 inactive receptors, I will still get activation of one of them during the next second, because this number did not change. But now this receptor that stays here has a chance 50% to stay in the active state. So probably the equilibrium will be a bit closer to the case when I have two receptors here, or something like that. So now each second one receptors come from here to here, being activated with the probability of 1%. And from these two receptors they have probability of 50% to be inactivated, so on average each second only one of them will be inactivated. The numbers may look small to you, but please note, by this stabilization we got from one active receptor on average to two active receptors on average. If we have some even better ligand, we can decrease this probability even more. So why don't we make this 1%? In this case, I can have 100 inactive receptors, and who will tell me how many active receptors to keep this more or less in equilibrium?
It's about equal chances.
Well, I guess 100 if there is 100, but yeah, maybe it will be better to write 50 and 50 on the both sides. Yeah, so we can have 50 inactive and 50 active. And this mechanism, or this idea of changing the probabilities, it basically gets through our course as we go. Because when we have considered chemistry, I often reminded you about stability of certain chemicals, or when, I don't know, some hydrogen goes away and whatever is left, some conjugated base, is it stable or not stable, and so on. That's basically about these probabilities. So there is some probability to change something or to stay in that state. And here you see when ligand, basically the same idea. Ligand binds to the receptor and decreases the probability of this receptor to deactivate. Or you might say it increases the probability of the receptor to stay in that state. So that's basically the meaning of that word. So that ligand is stabilizing the active state of the receptor. And you see how changing, how manipulating these probabilities, we were able to get of almost everything turned off. So 100% of receptors, 100 receptors were in state off and just one was in state on, to the state when we have 50 receptors off and 50 receptors on. That's quite many, because if you recall the numbers we had in this lecture, the concentrations of, let's say, of glucagon, they are much smaller than the concentration needed to activate 50% of all the receptors. So you just need a small portion of them to activate, to start some sort of information transduction and some sort of pathways, biochemical pathways. Good. Is this more or less clear?
Yeah, thank you. That was quite understood.
Good. Okay. I guess I ran out of time, so probably this is a really good place to stop. So thank you for being here today. Once again, congrats on getting to the 50% of the course, and I hope that we will stabilize the rest of the course and we will get through all of this. Good. And please consider maybe you want submit some midterm or something, because this is pretty simple course. Would be not good if you don't pass. I don't want that to happen. Good. So have nice evening and see you tomorrow.